Hello, everybody. Welcome to our penultimate colloquium this semester. We'll have one more after this one. A heads up that that's going to be Mariam Ali from Columbia University. She'll be joining us in a couple of weeks, April the 19th. Just a couple of housekeeping issues. As always, please feel free to put your questions into the chat box at any time during the course of this afternoon's talk. And we will leave time at the end for me to moderate those questions with Dr. Lee. And at the end of the formal talk at 1 o'clock, you'll be able, if you wish, to join us, what we like to think of as backstage, so to speak, in a more informal Zoom meeting, or actually Teams meeting, I should say. Still nostalgic for Zoom. You'll get the link to that in the chat during the course of the talk. So it's my great pleasure to welcome a new colleague, a new School of Behavioral and Brain Sciences colleague, Dr. Yune Lee, who is affiliated with the Kalia Center, will be based for his research up at the Kalia Richardson. Dr. Lee joined us as an assistant professor in August 2020, with all that might entail in terms of his opportunities to interact with folks and get his research up and running. We're hopeful that all this is coming to a rapid end, and we will actually be able to interact with Dr. Lee face to face, but we're very grateful to him for doing this virtual talk with us today. So Dr. Lee completed his PhD at Dartmouth University and went on to a postdoc at the University of Pennsylvania. He then joined Ohio State University as an assistant professor, and then joined us in, as I said, August 2020 as an assistant professor here in the School of Behavioral and Brain Sciences. And his interests are in the behavioral and the neural relationships between music and language, and how these relationships might be leveraged for translational purposes. And today he's going to give us a talk called The Brain, Music and Wellbeing, the BMW story. Without further ado, over to you, Yune.
Well, thank you so much for your very nice introduction. Okay, let me go ahead and get started. All right, so as many of you are aware of this tragic story of Gabby Gifford, who had a headshot back in 2011 when he gave a public speech. The breaking news initially said she got killed, but she miraculously survived. Although she lost her speech and language ability because this bullet went through her left hemisphere, which destroyed the language network. So as a politician, that could have been a devastating disorder. Now, this is a little video that I'm going to play where she's meeting with a speech therapist. So let me just play this video instead of me going forward.
You can do this. You ready? Little light of mine, I'm never let it shine.
Remember, at this point, she couldn't say the word light. Suddenly, there she was, singing that word and a whole lot more.
I'm gonna let it shine, let it shine, let it shine, let it shine. Excellent.
All right, so as you just heard, she had trouble saying the word light, but she was able to sing quite fluently. So this is almost 200-year-old observation. People with speech language disorder, it's called aphasia, they have trouble speaking, but when they are asked to sing, they can sing well. We don't exactly know how that happens, but based upon that phenomenon, in the 70s, people developed this technique called the melodic intonation therapy. So in this cartoon, what you're seeing here is the therapist and the patient, they're actually singing together while they're tapping along. So we used to believe that this melodic or pitch variation is the primary factor that could enable speech fluency. And this rhythm tapping is thought of as secondary. Now, but recent studies have actually suggested that rhythm is just as important as melody or pitch variation. So let me just briefly walk through this data here. So along the x-axis, we've got different lyric conditions, singing and then on the y-axis, we've got correct syllables after speech test. So as you can see here, between rhythm and singing condition, they're actually yielded pretty comparable results, suggesting that the rhythm is actually just important as important as singing. So I was quite inspired by that, and then I decided to develop this rhythm-based intervention program. So this program consists of 4 different steps. So in phase 1, well, we teach them tapping on, just tapping only, just rhythm tapping only, no speaking whatsoever. And then after they are familiar with this rhythm sequence and rhythm tapping, then we introduce a single word. And then after they get the hang of it with the rhythm tapping, then we gradually increase the number of words to the sentence or phrase level. And then finally, in the phase 4, they're trying to speak without tapping in a more naturalistic way. So this curriculum of activity actually has been implemented as a video gaming therapy. It's called the Speech Hero, and we recently got NIH SBIR award with this program, and we're about to use this for the upcoming clinical trial with another NIH grant. Now, let me describe this case study where we had a 62-year-old female native English speaker. So she had a left MCI stroke. As you can see here, there's a massive lesion on the left hemisphere. So that gave her severe broadcast-aphasia. She was completely non-fluent. For some reason, however, she was able to speak the word time, only that word time, very fluently and spontaneously. So when I met with her, people called her Time Woman, and I had no idea why. But she was using this time in a way to communicate with others. And I was very impressed. When I first met with me, she introduced herself like time. Instead of saying my name is Debbie, I was able to guess because her melodic prosody, everything, rhythmicity was all preserved. Now, so after she enrolled, we taught her several different phrases that are attached to different rhythmic patterns, such as I love you, as you see here in the music notation, like I need a drink, and so forth. And we did 8-week long pre-post testing in between the intervention. We also inserted this baseline condition, just wait 8 weeks so that that period can serve as control condition for herself. And then we had also MRI scanning in between. So let me play this video. So just watch this.
Open. Hi. 2 more. Hi. 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 Yeah. Wow.
That's the first time she said hi ever since she had a stroke.
You fixed it. You controlled the mouth. All right.
She was so happy, she became so emotional.
I'm so proud of you. All right, let's keep going. Ready? 1. There. There. I love you. I love you. Good. I love you.
So this is where the beta version of Speech Hero came into play.
Yeah, you're doing it.
Yeah, towards the end, she improved dramatically. The difference was day and night. Jamie is our daughter's name, so we decided to teach her that. So now you see she's no longer tapping.
I love you.
All right, so after the therapy, she, like I said, she improved greatly, and she was able to speak a total of 42 words across 16 different sentences. And she also showed some improvement in some existing battery tests, such as Western aphasia battery repetition result. You can see that over the course she improved, but the magnitude of improvement was actually greater between pre and post than between pre and baseline. Likewise here, for the naming word finding, she showed larger improvement after the therapy session. And this is MRI data, pre-post difference, diffusion tensor imaging. So on the right side, there's a pretty figure that show a bunch of white matter tracks. And we measure this structural connectivity change in 3 different areas, namely left inferior frontal gyrus, right inferior frontal gyrus, and the right posterior superior temporal gyrus. Because of the lesion, we were not able to get the RI from left superior temporal gyrus. Now, as you can see on the left side, in between the therapy session, you can see that there is an increase of structural connectivity change. So y-axis here, this is the index of structural connectivity. So you can see that there's an increase of connectivity in the right side, right hemisphere. However, if you look at the left side, there's a slight decrease. So obviously this is from one subject, but this suggests that the right side of the intact tissue, the homologue, they seem to take over the language function from the left side, which can explain her improved speech fluency. So we're planning to collect more data and conduct the NIH project now as the pandemic is coming down. All right, so here the central question, the key question is, can we better understand the neurocognitive and genetic mechanisms by which rhythm contribute to language? So over the next several minutes, I'm going to describe this schematic. So the connection between music and language, they're all heavily, they rely on temporal processing. And this temporal processing is mediated by this dopaminergic system, which is centered along the frontal striatal network. And obviously, there's a genetic influence to this. So let me go ahead and explain this. So what you see here is the activation of the brain network when we give some language grammar task. So you can see the conventional left lateralized Broca's and Wernicke's area. And also you see this bright activation in the middle of the brain that's called the basal ganglia. So that's co-activated when you do this grammar task. Now, what you see here on the bottom is the areas that are activated when people are doing some kind of rhythm task. And you can see this area, the basal ganglia in the middle, they're activated quite a bit. So you can tell what areas are actually overlapped with each other. Now, I'm going to highlight this area again, the basal ganglia, especially this striatum right there, where there's a lot of D1 and D2 dopamine receptor. You can see the red color right there. And on the right side is the dopamine receptor cartoon right here. Is a cleavage that's binding site for different dopamine subtypes, and which is determined by this inhibitory G protein. That's a little bit of biology stuff. Anyway, so there is this gene called the DRD2 gene, and if you have a polymorphism of that gene, meaning that one nucleotide is swapped with others, it's called the SMP or single nucleotide polymorphism. And the consequence for that DRD2 polymorphism is that there is a great reduction of D2 receptor in the striatum that I just described, highlighted. And it behaviorally manifests itself as poor rhythm timing ability and also poor grammar ability, interestingly enough. And then also it has been implicated as a poor working memory. Now, I'm going to describe this little study, which was done by my friend, colleague. We were the office mate back when we were at UPenn. So in this study, he brought Penn undergrads mostly, and then he asked them to move their fingers just at their own comfortable tempo. He never asked them to move the finger fast or slowly, just whatever tempo you find most comfortable. And then, so that's what's called the spontaneous finger tapping task. And then he went ahead and genotype subjects on the basis of the following 3 genes. One of them is obviously DRD2 gene. Another one is COMT gene that is also implicated as a dopamine, as many of you may know, and as a control, BDNF gene. So what you see here is on the y-axis, that's a mean inter-tapping interval. What that means is how long it takes from one tapping to another, and he took a mean average. And as you can see here, he only found a significant difference between people who have DRD2 polymorphism and people who don't. He didn't find such pattern in other genes. So what that means is that the people with the DRD2 polymorphism, they inherently, they're just slower in this rhythm tapping. This rhythm tapping processing is somewhat compromised, and could be because of this DRD2 polymorphism. So as a follow-up study, we did the fMRI study where we gave them a simple timing task. So I'm not going to go through all the nitty-gritty, just hit the take-home here. So the sub-second is harder timing task, and the supra-second is tend to be easier timing judgment tasks. So we gave them 2 dots. They appeared sequentially, and then subjects had to judge whether the duration is the same or different. And as a control, we gave a call task to control for visual differences. Now, so in the sub-second, you can see that in this basal ganglia striatum, you can see this difference between sub and supra-second, only in the polymorphism group, but not in the control group. So we published that paper a couple of years ago. Now, as we were writing up that manuscript, we stumbled upon a very, very similar study. They also did fMRI study. They were looking at the DRD2 gene. So it was almost identical, except for the fact that they were looking at the consequence of DRD2 on the grammar learning task. So they taught subjects some synthetic language, and they measured their grammar performance. And this is a behavioral data where they show that actually A1 is a polymorphism group. They perform poorly than control. And as you can see here also in the basal ganglia, there's a big difference between the 2 groups. So when I first stumbled upon that, I scratched my head a little bit because I was not quite clear about the connection between grammar and the rhythm and this dopamine, the DRD2 gene. So, you know, that was my initial curiosity. And then I finally had a chance to actually see if there is a direct connection between people's rhythm ability and their grammar performance and language ability. So we brought a bunch of actually children group, and then we gave them 2 simple tasks. One is rhythm discrimination task, another one is language task, where the syntactic structure was manipulated. And we found that kids with a better rhythm discrimination ability, they actually perform better on this language task. And that effect remains significant even after we control for all other demographic or even working memory aspect. So that was published last year. Now, so this rhythm is actually, the rhythm deficit is quite prevalent in dyslexia or SLI, specific language impairment individuals. So they actually show not only impaired rhythm perception, but also impaired rhythm production. We still don't exactly know, but I'm trying to test out this theory with this model. So whether it's a rhythm in music or grammar in language, they're connected by this shared temporal processing network, which is mediated by this dopaminergic system. And if you have any mutation in that gene, that this whole cascade of processing is going to be affected. So this is a neurogenetic mechanism that I have been and I'm going to continue to test over the next decade or so. Now, when it comes down to dopamine, it's hard not to think about this particular neurological disorder called the Parkinson's disease. And this is a PET imaging data. You can see that between the two normal and Parkinson's group, there's big difference in terms of their dopamine intensity. So this is a way of highlighting the level of dopamine, and this is a way of diagnosing whether someone's got Parkinson's disease or not. Now, many people know that the Parkinson's patients have trouble moving their body. They have a lot of tremor. They also are heavily depressed, but there are not many people actually know that they also show speech and language impairment. So I'm going to describe this study done by German group, where Parkinson's patients were brought up, and then they were presented with a bunch of short German sentences, and half of them are syntactically correct, and the other half are syntactically wrong. So, for example, instead of saying, I want to eat pizza, it could be something like, I want to pizza it, something like that. And then you can tell that actually they're grammatically wrong. And this is the EEG profile right here. So whether a Parkinson's patient was presented with syntactically correct or wrong, their EEG pattern look more or less the same. Now, the same Parkinson's patient is now primed by music that has a very heavy rhythmic pattern. The rhythm part is heavily emphasized, such as marching music, like a guang, 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 for a few minutes. And then they perform this task again. And now you can see that, you can see this P600 component, which is associated with a syntactic violation. In fact, this P600 is the hallmark of a syntactic processing in EEG. So you all of a sudden see this P600 associated with the syntactic violation. Now, they went ahead to see if this is a general music effect or this has to do with music with strong rhythm. So they played another type of music as a prime, and then the same subject did this task again, and you can see that this P600 is no longer there, it's gone. So they concluded that this rhythm is so important to temporarily improve their grammar ability. Now, inspired by this recent study, I devised this new rhythm therapy program for Parkinson's patients. So I termed this DDR class. So DDR is, used to, will still stand for dance dance revolution. I'm sure you know that, but here DDR actually stands for drum dance rehab. So we offer this class to a couple of Parkinson's patients at the beginning as a pilot study. And the idea is to combine the two most well-known and effective rhythmic exercise, namely drumming and dancing. Okay, so when they came to the class, they followed the instructor's guide, and then they play drumming together in synchrony, and they dance together. So I have a little video here. See if it works. I can show you maybe a 20 second or something.
Yeah.
Okay, so hopefully you got the idea now. And go back to my slide. Okay, everybody sees my screen again, hopefully, and I hope that gave you some good idea. All right, so we measured their music, language, and working some cognitive ability before and after this intervention. We offered this 5 days a week and 3 weeks long, very short but intensive, very intense program. And you can see that they all pretty much show improvement. And you can also see there's a huge individual differences, but regardless, they show quite a bit of improvement. And as we were doing second DDR project class, we were hit by the pandemic and we couldn't finish the project. All right, so before I end my talk for the next few minutes, I'm going to touch upon this this study that I'm going to start soon. So there's another great example of a neurological disorder, namely Alzheimer's disease, that get greatly benefit from music. And I'm sure that you've seen some anecdotal videos or stories where music all of a sudden invokes someone's nostalgia and their memory associated with that music. And that's why music has been quite commonly used in some assisted care place or some family people. Actually, they use this music to help out their parents or their loved ones. So there has been some studies systematically comparing what kind of music actually is the most effective. And as I'm sure many of you know, this Mozart effect, there is a big fad about the classical music, Mozart music, but turns out to be that's not the magic bullet. People debunk this theory. In fact, what is most effective is the music that the patient, the individuals love the most, their all-time favorite music. So it doesn't matter what kind of genre that is. What matters is the music that this person is attached to for several decades. So that study recently came out. Now, so in this study, we're not just playing or providing music that the person loves, but also this music is going to be processed by being combined with a specific frequency. It's called the binaural beat frequency. For those of you who are not familiar with this, so this binaural beat is created by playing two different frequencies. For example, 440 hertz on the right side of the ear, 400 hertz on the left side of the ear. And the brain is experiencing some confusion because there's a discrepancy in this their frequency they're hearing. So what the brain does is to generate the third new frequency that corresponds to the difference between the two. So in this case, the 40 hertz that is generated by the brain, and that is in turn entraining some neural oscillation of certain brain modules. So there is a growing awareness and a growing number of studies showing the benefit of efficacy of this binaural beat. So binaural beat has been shown to improve mood, emotional regulation, or attention and memory. Although there's very few neuroimaging studies to show some neurological mechanism or neuronal mechanism there. Now we're choosing this gamma frequency binaural beat, which is a 40 hertz. So why 40 hertz? Recently, there is a study done with a transgenic mouse model where this mouse was exposed to this 40 hertz flickering sound or light, and 1 hour per day, 7 days a week, just 7 days long, that's all. And then they found out that this gamma frequency range, which is initially looking very funky in the mouse, transgenic mouse, that loop becomes normal. And also that is associated with reduced amount of amyloid beta, which is hallmark of Alzheimer's disease. And to my knowledge, there's no human study actually showing this yet. So here the idea is to combine, again, the personalized music, the music that any individual loves the most, combined it with 40 hertz beat, binaural beat. And this is what I called Therabeat. And this Therabeat app was recently chosen as UT Dallas senior capstone project, and a group of students, actually there are two teams, they have been working so hard to develop this app during this semester. Now, once we develop this app therapy, then we're going to actually do little clinical trial. I submitted a few grants for this, and so we're going to do this pre-post testing, obviously in between the therapy, and we encourage patients to listen to this 1 hour per day, 5 days long, and 3 months total. So the idea, and then we're also going to do neuroimaging, collect some brain data using MRI machine and using EEG device, so that we can track this spatial temporal pattern. Now, and then we hypothesize that after this therapy use, well, we might be able to see this increased gamma band of an EEG signal, and just like what the animal study has shown. Also, we might be able to see some increased functional connectivity in the memory network comprising both anterior and posterior part of the middle temporal lobe. And by contrast, in the control group, where they probably don't show such spatial temporal profile. And most importantly, we hope to see that patients will be happier and they show increased well-being score after this therapy use. And longer term goal is obviously to disseminate this therapy app to the wider Alzheimer community. Now, so far I've shown you some studies where music can be used as a therapeutic regimen, could be a very effective therapeutic regimen, by talking about my ongoing and upcoming projects. And over the last decade or so, there is an increasing effort of better understanding how music works through this multidisciplinary approach. And to further fuel that momentum, NIH has stood up, and they recently funded several music research, including my own. And then also they have been sponsoring this Sound Health Network, which is quite active these days. All right, so with that, I want to thank my research team and my funding agencies, and I want to thank you for your attention.
Fantastic. Thank you very much, Yune. We have plenty of time for Q&A, and then just to remind people, we can log on to another meeting, another event subsequently to talk about things more informally. So we have a few questions. Let me see if I can moderate these. So with regard to the clinical trials that you were describing, obviously a crucial question is what the control group is and what the control interventions are. And I was just wondering, what are your thoughts on that? With regard, for example, to the Parkinson study, what is the appropriate control condition against which to assess the efficacy of the drumming intervention?
That is a great question. And like I said, we were about to do larger scale, full-blown study with a Parkinson patient to confirm this effect of DDR. And we're going to actually offer normal, regular exercise class, which affects systematic rhythmic movement. Yeah. So you can control this movement and motor functioning, but then it just lacks rhythmic process.
Yeah, that sounds really cool. What about for the Alzheimer's study? What would be what is the control intervention for the therapy study?
That's a great question. I had to think a lot hard to figure out, would the music without binaural beat be a good control group, or offer them some other cognitive therapeutic regimen. Right now, because again, we're still we haven't started. We're still considering this idea. Right now, we're planning to just do this passive doing nothing group, and then see if that is actually see if the therapy still shows some improvement compared to this passive listening group.
Right.
So
when you say passive listening, you mean encouraging people to listen to music for the same period of time as they, but without the superimposed beats.
So that will be active listening, actually.
Oh, okay.
But passive, I mean they doing nothing. So do nothing group.
Okay. But that's probably not a great control, is it? Because people generally improve when you pay attention to them.
Exactly.
Yeah.
Right, I agree. So we're thinking of, still we're in the middle of brainstorming, but active listening or some other cognitive exercise could work. Yeah.
Very cool. Yeah. So I have a question here with regard to the relationship between the very, very interesting results, I think, between language development and rhythmic ability in children. Obviously, you're trying to generate a causal model, and you showed us a kind of nascent model. But there's no reason, on the basis of the available data right now, is there to think that the causality goes from rhythm to language? Could it not be that children who are more adept at learning language are the ones who also acquire better rhythmic ability? In fact, the causality could go in either direction.
Yeah, that's another great question. So we're actually not testing causality per se. We're testing this shared temporal processing network that is mediating both music and language processing. And we're actually right now planning to do the causality testing by doing this simultaneous TACS and fMRI study. So I've been talking to the vendor. We're interested in facilitating this sensory motor network, including SMA and premotor, using beta frequency beat, because that frequency range has been implicated. Mm-hmm. And then see if that could improve their not only language ability, but also rhythm ability. Mm-hmm. So yeah, we're actually planning to do that.
Interesting. So someone has just raised this question about 40 hertz rhythm. I'm just going to expand that a little bit. 40 hertz from a EEG point of view, that is very low gamma. So I'm sure you appreciate there's gamma activity goes up to like 120 hertz. Mm-hmm. And in fact, quite a lot of people have argued that low and high gamma are functionally distinct and probably have different neural underpinnings and different functional significances. But why would one think there is something magical about entraining low gamma, given the very large bandwidth of gamma and the controversy over which frequency bands are the ones that are most important, if you like, for understanding neural function?
Yeah, that's an excellent question. It is actually very difficult and hard to choose carrier frequency because it's somewhat arbitrary. Yeah. And we chose this 40 hertz based upon literature where animals show very funky looking shape 40 hertz gamma range frequency when they did this electrophysiology. Yeah. And as you point out, gamma is not this. Gamma is very heterogeneous. There's a low band gamma, high, mid, and high band gamma. All of them have been associated with implicating different cognitive processing. And in fact, low gamma is somewhat overlapped with high beta. Yeah. So it is actually quite tricky problem, but we try to base our decision on the existing literature.
I understand.
There's certainly room for exploring other bandwidth.
Yeah. Yeah. I think that would be a very interesting study, actually, would be to look at whether the carrier frequency or whether the resulting frequency that you get, whether it's 40 hertz, 60 hertz, whatever, how much of a difference that makes. I think that would be a fascinating study.
Yes. In fact, I did have a chance to show our pilot data that we just began to collect as of this semester. And we saw some promising data out of this beta range, and we're about to test out higher gamma frequency or even lower theta level. So hopefully I'll be able to show you more data later on.
That's great. One last question at the moment, if no one else brings one in, but this is going right back to the beginning of your talk. What do you think is the mechanism that gives rise to the changes in DTI parameters as a function of the therapeutic intervention that you were describing? Why should, what is it that makes, given what we know about myelin, axonal integrity and the like, why should you see an increase in FA times MD as a function of this kind of an experience?
Yeah, so there is actually a great debate about the role of contralateral hemisphere. One of my colleagues called this good, bad, and ugly. What that means is we actually don't exactly know the underlying neural mechanism, and it's all there's a lot of individual differences. So we think that what matters is the size and configuration of the lesion in the left hemisphere. So depending on that, some patients might develop plasticity within the ipsilateral area in the remaining healthy tissues, and they develop some new myelin connection and so forth. Whereas some patients, like my patient, the Thai woman, who had a massive lesion on the left hemisphere, there's not much left. As I show you, there's no superior temporal gyrus there. So the brain probably choose to utilize the contralateral side through the connection of a corpus callosum, and then it starts to mediate speech-language fluency or speech-language function. And we know that actually right side playing more dominant role in music processing. Mm-hmm. So this very intense music activity, especially rhythm, might really promote the brain plasticity on the right side, and then use this remaining healthy frontal striatal network. In my study, her basal ganglia on the right side is also very intact. So it really depends on it. It could be case by case. So that's where the difficulty of this aphasia lesion symptom mapping is at.
Very cool. I'm not seeing any more questions now. So I'd like to encourage those who would like to do so to join us in our chat room. We can continue a little bit more informally and have the opportunity to interact with Yune face to face and try to work ourselves slowly into the thought that one day we'll all be in the same room. So thank you so much for a tremendous talk. Very, very interesting work. Very, very glad to welcome you to the BBS community, and we look forward to hearing about your research going forward. Very exciting. So thank you very much, Yune, and I hope to see you in just a couple of moments on our other URL.
Thanks so much, Mick.